Now, getting into the lecture today, the topic is biochemical and technical concepts in the Seahorse XFP cell mito stress test and real-time ATP rate assay. Mitochondria do a lot of things, and we might want to measure their performance and their ATP production. But how can we do this? Well, as a general concept, it's possible to use compounds to selectively alter mitochondrial protein function, and by measuring mitochondrial oxygen consumption and extracellular acidification, we can infer how different aspects of the mitochondria are performing and how much ATP they're producing. It's possible to build your own rig to do this with an oxygen sensor and a pH meter, but it is easier and more efficient to use the Seahorse XFP or related instruments and the reagent kits that go with it. We have one of these on campus in the stem cell core. There are other Seahorses that exist that you can use. I believe there's an XF24 at Loma Linda, which can be used for tissue samples. So you're not necessarily limited to just this instrument in particular. But if you do decide to use this, it's important to be able to understand the concepts associated with it so that you can interpret the data. I'm going to start with a very brief review on the Krebs cycle, electron transport chain, and ATP synthase, proton flux in the mitochondria, what we can measure in the mitochondria, what the Seahorse XFP is, and I'm going to talk about these two assays right here in detail and the data you get from them. So mitochondria perform many functions in the cell, not all of them necessarily related to ATP production directly, but ATP production is the main thing that they do. We know they turn pyruvate and fatty acids into oxygen, ATP, water, and carbon dioxide. This makes up respiration. In particular, I'd like you to pay attention to and note the consumption of oxygen and the production of carbon dioxide in this process, as that will become very important later. This is carried out by the Krebs cycle, also called the citric acid cycle, and it takes place in the mitochondrial matrix, which is inside the inner membrane. Specifically, once this cycle allows NADH to produces NADH, and this is then used to hand off electrons between different protein complexes in the electron transport chain. And this pumps protons from the mitochondrial matrix into the intermembrane space, creating a proton gradient, a difference in the amount of protons between the mitochondrial matrix and the intermembrane space. That process consumes oxygen, and then the protons re-enter through ATP synthase, and this creates ATP, which then leaves the mitochondria and is used by the cell for other processes. Collectively, this process makes up something called proton flux, and we can quantify it by measuring the oxygen consumed by the electron transport chain indirectly. The cell looks to maintain a constant proton gradient and constant ATP levels in the cell. So it is reasonable to assume that the cell will do this, and that will manifest as a change in oxygen consumption consumed by the citric acid cycle and the electron transport chain. And so by modifying this, measuring oxygen consumption, we can infer how different aspects of the cycle are performing. The Seahorse allows us to do this. And specifically, the two variables that we are measuring in the Seahorse are pH and oxygen consumption. That's essentially what this device is. It is basically a very expensive pH measurement device and oxygen sensor. And it also allows us to treat our cells with different compounds. So let's talk about the first assay, the Mito stress test. There are a lot of assays you can use on the Seahorse. The Mito stress test and the ATP rate assay that we'll be talking about later are the ones you'll see most frequently in publication. There are some other ones as well, but this Mito stress test is very popular because it allows for the measurement of a fairly large number of parameters. Specifically, you can measure the basal respiration of your cells. You can measure the oxygen they devote to ATP production. You can measure the maximum respiration and their spare capacity, non-mitochondrial oxygen consumption, and proton leak. And I'll be talking about each one of these in detail in just a few minutes. But I'd like you to note in this graph, it's not possible to get all of these parameters until the complete assay is completed. So you don't necessarily get proton leak or non-mitochondrial oxygen consumption until the very end. So there's no real good way to talk about these in sequence, but I'm gonna try to have them in an order that makes some sense. So the first measurement that we're going to talk about is basal respiration. This is the amount of protons being pumped across the membrane under normal conditions. When the device measures this, you're just measuring the cells before they've had any compounds added to them at all. And the reading you get is the picomoles of oxygen consumed per minute per whatever normalization factor you're using. Say, in our case, we use number of cells. So that's this reading here. Now, this is not the amount of oxygen devoted to ATP production. This is the amount of oxygen that the mitochondria are using via the Krebs cycle to pump protons across the membrane. Those protons might be used to generate ATP, or they might be lost to proton leak, which I'll talk about later. And so what you will get from this is you'll get 3 data points on the Seahorse. The Seahorse will give you 3 data points taken over 15 minutes of what this reading is, and this is what the data looks like. It's oxygen consumed in picomoles per minute. The second thing that this assay allows you to measure is ATP synthesis. This is a measurement of the picomoles of oxygen per minute that the mitochondria or cells are using to produce ATP specifically. Now, this is different from the measurement before because this does not include the oxygen used to pump protons across the membrane that are then lost to proton leak, which is a different concept. This is achieved by treating the cells with oligomycin. It shuts down ATP synthase, and when this occurs, the cells can only re-enter the matrix through proton leak, as you see here. So you get 3 additional measurements under this condition, and that allows you to infer the extent to which the cells are consuming oxygen to produce ATP. The next 2 measurements you get are measurements that are closely related: maximal respiration and spare capacity. This tells you the maximum amount of oxygen that the cells can consume to pump proteins across the membrane overall. What's the hardest the cells can work, the most oxygen they consume. You get this reading here, and it also tells you how much spare capacity the cells have to pump protons across the membrane. And this is achieved through treating the cells with FCCP. You have to actually titrate this to determine what the best dose is for your cells. But once you do that, FCCP is a protonophore that allows protons to cross the membrane without passing through ATP synthase. And in this case, you can get a good idea of the hardest the cells can possibly work to pump protons across the membrane. And the reading you get in terms of oxygen consumption after FCCP treatment is typically a lot higher. So here you can see after that treatment, the oxygen consumption in this example goes up by a lot. Having obtained this, the next reading that you can get is non-mitochondrial oxygen consumption. In order to get this, the cells are treated with rotenone and antimycin A. Now, these 2 compounds shut down respiratory complexes 1 and respiratory complexes 3, respectively. When this occurs, the electron transport chain is shut down. So no protons are being pumped across the membrane. And so in this case, when this occurs, you can measure the drop in oxygen consumption. And you can see here in this example graph, oxygen consumption is decreasing very quickly after treatment. And so whatever oxygen is remaining, you can attribute to non-mitochondrial oxygen consumption. Because in this case, the mitochondria and the Krebs cycle are not consuming oxygen. Anything that's left is going to one of these other processes. Now, what sort of processes use oxygen that are not mitochondria? Oxidase enzymes would be one example. If you're dealing with cells that might respond to an infection, there are proteins on the cell's surface that produce superoxide radicals. That I believe is one example. Lysyl-like oxidase enzymes are another. Loxyl-1 uses oxygen to produce to catalyze its reactions, as you can see here. So that is another potential source of non-mitochondrial oxygen consumption. There are others as well that you can read about, but this is just an example of the sort of thing that you might expect to observe consuming oxygen that are not mitochondria. Finally, we have proton leak. As I mentioned earlier, this is the amount of protons that cross the membrane without going through ATP synthase. This can occur a number of ways. They can cross directly if the membrane is, but they can also pass through specific proteins in the mitochondrial membrane that allow this. There is evidence that this can be part of a deliberate process to regulate reactive oxygen species consumption. Now, this process consumes oxygen because the protons must be pumped across the membrane by the electron transport chain beforehand, but it's not generating ATP by definition. So when you complete one of these assays, this is the kind of readout you're going to get. In this example, the blue is the control group, the orange is the treatment group, and you can see here it follows that same pattern. Here are our basal readings. Oligomycin is administered here. Oxygen consumption drops. Then FCCP is administered. Oxygen consumption increases. 3 additional readings are taken. Finally, rotenone AA is administered, and oxygen consumption drops again. And overall, the data you'll get is something like this. You will get basal respiration, proton leak, maximal respiration, like we talked about, spare respiratory capacity, non-mitochondrial oxygen consumption, ATP production, and efficiency measures in terms of their spare capacity, and coupling efficiency, which measures how closely ATP production is coupled to oxidative phosphorylation and pumping protons across the membrane. All of these readings are in picomoles of oxygen consumed per minute per whatever normalization factor you have. So, like I said, if you're doing per 10,000 cells, which is what we do, a lot of people do per 10,000 cells, then that's what this data is going to give you. And I want to reiterate, this data here is ATP production. This isn't the amount of ATP produced. This is the amount of picomoles of oxygen per minute that the cells are using to produce ATP. But let's say you want to know the actual levels of ATP produced specifically in the cells. For that, there's another assay called the ATP rate assay. Now, this is very similar in concept to the Mito stress test that we just talked about, but it differs in some ways that I'm going to talk about, and it allows you to quantify the amount of ATP the mitochondria produce, and it also allows you to quantify the amount of ATP produced from glycolysis, and it can allow you to do this if you normalize per cell in an absolute way. So you can quantify the absolute picomoles of ATP from each source produced per minute per cell if you're normalizing based on individual cells. So here's how this works. Much like the Mito stress test, 3 measurements are taken of oxygen consumption and critically extracellular acidification as well. That's that pH measurement that we talked about. Now, these are just the basal readings just to see what normal readings are under normal conditions. This graph here is showing OCAR, like you saw in the Mito stress test. Then cells are treated with oligomycin, just like in the Mito stress test. Because ATP synthase is shut down, the fewer protons re-enter the matrix here, and so the cells and the mitochondria pump fewer protons up into the intermembrane space. Remember, they're working to keep that constant. So if you reduce the amount of ATP that's re-enter the amount of protons that are re-entering the matrix here, the cells will recognize that. They'll recognize the potential here is going up, and they'll pump fewer protons across the membrane. So it's reducing oxygen consumption indirectly. That's what's happening here. And under these conditions, protons are only re-entering through proton leak. So 3 additional measurements are taken after this treatment. However, unlike the Mito stress test, there's no FCCP administered. Rotenone and antimycin A are administered. When this occurs, the electron transport chain shuts down completely. No oxygen is consumed in the mitochondria, and overall, and so this treatment is important because under this condition, the only thing contributing to the acidification of the media the cells are in is going to be glycolysis. So although pH is being measured this whole time, because carbon dioxide acidifies the media the cells are in, you're not going to be able to know necessarily how much of that acidification is from glycolysis versus oxidative phosphorylation producing carbon dioxide. In particular, let's take a closer look at how extracellular acidification is relevant. So glycolysis is another source of ATP production in cells. This process produces pyruvic acid and protons, which are acidic. This makes the media the cells are in more acidic. And so by measuring pH, you can infer how much of this process is going on, how much glycolysis there is, and then you can infer how much ATP the cells are producing from glycolysis. However, carbon dioxide from oxidative phosphorylation also contributes to this. So by shutting this down, all that is left is glycolysis occurring. That's the only thing making the media more acidic. You measure the rate at which the media gets more acidic. You can infer the level of glycolysis. And so here, this graph shows you what happens to extracellular acidification rate under treatment with oligomycin and rotenone AA. In particular, you can see here, after oligomycin treatment, the rate at which the media becomes more acidic increases. The cells are performing more glycolysis to produce ATP since they can't produce it through ATP synthase because oligomycin shuts that down. Then rotenone AA, rotenone antimycin A shuts down the electron transport chain completely. So you do not even have you don't even have oxygen being consumed and carbon dioxide generated from oxidative phosphorylation, and you don't even have that making the media more acidic, and that really allows you to infer just how much the cells are relying on glycolysis to produce ATP. And at the end of this assay, you get something that looks like this. You get the picomoles per minute of ATP per number of cells produced from each source. So in this graph, the red indicates the amount of ATP produced from glycolysis. The blue indicates the amount of ATP coming from mitochondria under the control. And this group here, 100 nanomolars dexamethasone, this is the treatment. And so in this data, we would see that our treatment here causes a decrease in ATP production from both mitochondria and from glycolysis. And so this assay really allows you to see exactly what's happening in terms of ATP production in your cells. There are other assays that you can perform on the Seahorse, the glycolytic rate assay kit, the glycolysis stress test, and a few others here. Generally, the ones I just mentioned are the most common you'll see published, and they're the most common here on campus. There are additional technical details that I'll be talking about later. I'll be recording that as well. But if you want to learn more today, there are videos, training, and support available from Agilent, the company that makes the Seahorse. And with that being said, I want to open it up to questions at this point.
Hi, Shane. Nice presentation. So you talk about when you normalize these data, typically a typical way to do this is to normalize to the amount of cells, right? So then the cell counting becomes very important in terms of getting an accurate number, right? So what is your strategy in terms of getting the reproducible or accurate cell number?
So what we do is we stain our Seahorse plates with hoaxed. We add the hoaxed directly to the wells without actually rinsing the plate. So once the plate comes out of the Seahorse, no media is ever removed from it at all. What I do is I add a concentrated hoaxed to the wells. It takes a little while, but it does stain the nuclei. We then image it on a fluorescent microscope, and we count the cells in MATLAB. In particular, because sometimes nuclei can appear to overlap in the images, particularly if you have to use a very high density of cells. The computer can have a hard time distinguishing two nuclei that are close together from just one nuclei. So we have developed a custom algorithm that can actually count nuclei that appear to be overlapping. So what it will do is it will figure out what the average intensity per nucleus is. It will look at all of the objects, so all of the hoaxed stain in the image, and it will identify whether any objects is one nuclei or more than one. And if it's more than one, it'll calculate how many there are in that group based on total intensity. And I'll be discussing this in detail in the next presentation that I give. In terms of normalization, other people, I believe I've seen people do qPCR to normalize their wells. Some people do total protein content as well, total DNA content. But what you're seeing a lot of now is people actually counting the cells individually, and that's what we do. And the Seahorse that we have, there's a range within that it can actually measure oxygen consumption accurately, and it's in absolute terms 20 to I think 120 picomoles of oxygen produced per minute. And so with any of the readings you take, you want them to be within that range. And depending on how metabolically active your cells are, you might have to add more cells or fewer to keep it within that range. And so part of the process is optimizing the number of cells that you use. However, I know that your lab uses HeLa cells, and HeLa cells, I believe, are well documented. So you might be able to even skip that step and get a usable result right away.
Thank you.